The cell membrane, or the plasma membrane, is a thin, semi-permeable membrane that surrounds the cytoplasm of a cell. Its function is to protect the integrity of the interior of the cell by allowing certain substances into the cell while keeping other substances out. There are many parts to the cell membrane that are fluid, which is why it is called the fluid mosaic model. The macromolecules used in the cell membrane are lipids, proteins, and carbohydrates. The cell membrane is made up of phospholipids. Phospholipids consist of a glycerol molecule, two fatty acids, and a phosphate group that is modified by an alcohol. The phosphate is the phospholipid head. It has a negatively charged polar head and is hydrophilic, which means that it has a tendency to mix with and dissolve in water. The phospholipid tails are fatty acid chains that are uncharged and have nonpolar tails that are hydrophobic. This means that the tails do not have a tendency to mix with or dissolve in water. The cell membrane is amphipathic, which means that it has both hydrophobic and hydrophilic tendencies because of the phospholipid components. The phospholipid tails and heads make up the double layer of the cell membrane, with the cells being the inner region and the heads being the outer lining. The cell membrane is also a fluid mosaic model, so it is made up of many different proteins. These proteins are both integral and peripheral. Integral proteins are a type of membrane protein that is permanently attached to the membrane. It composes a significant fraction of the proteins that are encoded in an organism's genome, like protein pumps and channels. Peripheral proteins are proteins that adhere only temporarily to the biological membrane, with which they are associated with. They attach to the integral membrane proteins and penetrate the peripheral regions of the lipid portion of the bilayer. An example of this protein would be insulin pumps. Another component of the cell membrane and the phospholipid bilayer are carbohydrates. These carbohydrates are covalently linked to proteins, like glycoproteins, and lipids, like glycolipids, and function as adhesion and address the loci for cells. Cholesterol is in the membrane of animal cells. This lipid is in charge of maintaining the cell's integrity and plays a role in facilitating cell signaling, which lets our cells communicate. It also helps maintain the fluidity of the phospholipid bilayer by separating the fatty acid tails so they will not stick together and crystallize.
Cell membranes have a wide range of functions aside from just being selectively permeable. These other functions are mainly carried out by the proteins in the cell membrane. Hormone binding sites are where hormones and neurotransmitters and other chemicals attach to the surface of the cell. The receptor sites on a cell membrane leads to different effect on the different hormones. Similar to how certain enzymes only match up with certain activation sites, hormones and neurotransmitters are specific to each binding site. Different cells have different number of binding sites for the hormones and neurotransmitters. A real-world example is when insulin is secreted by an endocrine cell. This hormone will go out of the bloodstream, and if a cell doesn't have any insulin receptor sites, the insulin will not have an effect on it. The cell with the insulin receptor site, which is also known as the target cell, will be affected by it. The hormone-binding receptors are integral proteins. Both integral and peripheral proteins can act as enzymes and as a catalyst to speed up chemical reactions. Enzymes are proteins, and they make a biochemical reaction more likely to proceed by lowering the activation energy of the reaction, thereby making these reactions thousands or even millions of times faster than they would be without a catalyst. Enzymes in the membrane perform similar functions as those in the cytoplasm of a cell, as they transform a molecule into another form. A real-life example of this is globular proteins, which act as enzymes in the cell membrane. Cell-to-cell communication is also referred to as cell signaling and is vital for the function of the group of cell known as the tissue. Cell signaling is used to detect and respond to cues in the environment, and it allows for coordination among the cells. A real-life example of a tissue is a brain tissue or bone tissue. In adults, as the bones grow, the cells need to communicate through signals to sort themselves out. This communication is carried out by integral proteins. Proteins that are known as surface protein markers are embedded in the cell membrane and identify the cell. This enables nearby cells to communicate with each other. Cell membranes often include receptor sites that are used for the interaction with specific biochemicals such as certain hormones, neurotransmitters, and immune proteins. In this way, the cell can recognize and process some signals received from the extracellular environment. Cell signaling is also important for the balance between cell growth and death, as apoptosis, or programmed cell death, is triggered by the signaling between the cells. Adhesive molecules allow the cells to maintain their contact with one another and with other structures in the extracellular matrix. Integrins are the most important type of adhesive molecule. They act as mechanical links by relaying signals both to and from cells, and as a result, they can play an important role in sensing the environment and controlling cell shape and motility. There are also other types of adhesive molecules that are used to maintain physical contact of the cells. A real-life example of this is in epithelial cells, which line the inner and outer surface of the human body. The epithelial cells are a very good example of the different types of cell-to-cell junction. Protein complexes that are found within each cell further connect the transmembrane adhesive proteins to the cytoskeleton. The adaptor complexes bind adherens junctions to cytoskeleton actin, and other adaptor complexes bind desmosomes to intermediate filaments. This binding gives the cell its mechanical support, and it requires cell communication in order to be efficient.
Channels are proteins that are used in facilitated diffusion. These channels are glycoproteins, meaning they are proteins with carbohydrates attached, that allow molecules to pass through the membrane. These channels are almost always specific for either certain molecules or a certain type of molecule, like an ion channel, and so they are tightly linked to a certain physiologic function. A real-life example of a protein channel is GLUT4, which is a glucose transporter found in fat and skeletal muscle. Insulin triggers GLUT4 to insert into the membrane of these cells so the glucose can be taken in from the blood. Since protein channels are found within the cell membrane and not attached to it, they are integral proteins. Protein pumps are used in active transport, meaning ATP is required. In active transport, the molecules are moving against their gradient, making it harder to transport them. This is why protein pumps are necessary, because they use ATP and make the process more efficient. An example is a sodium-potassium pump that maintains resting potential in the cell. This protein uses the energy released from ATP to pump the 3 sodium ions in and out of 2 potassium ions into the cell. Similar to the channels, the protein pumps are inside the cell membrane, meaning they are integral proteins.
In conclusion, the cell membrane has many different components and functions that are vital for everyday life.